Good afternoon, ladies and gentlemen. It's my pleasure today to introduce our speaker, who is Robert Sapolsky, who is the John A. and Cynthia Fry Gunn Professor of Biological Sciences at Stanford University. He's also Professor of Neurology and Neurological Sciences, and he may even be Professor of other things as well, for all I know, since he tends to become a professor of lots of things, and with great justification. I think Robert Sapolsky is probably known, at least by name, to most of you here today, both as a scientist and as a great communicator of science. In science, his work in stress and the relationship of stress to human illness, which goes back to his days as a PhD student in Bruce McEwen's laboratory at the Rockefeller University, that work is at the forefront of its field. And he's clearly going to talk around that today. But it's also as a great communicator of the nature of primate society, including our own human society, that he's also known very widely and broadly. And this goes back much earlier than his PhD to his days as an undergraduate at Harvard, when he became interested in primatology, even to the extent, I understand, of learning Swahili, so that he could go to Kenya to study baboons. In baboon society. And he's continued his association with Kenya. He's now currently an associate of the National Museum of Kenya, and he's in Kenya on a regular basis to study his much-beloved baboons. He's written a number of books around this subject, which include the titles Why Zebras Don't Get Ulcers, The Trouble with Testosterone, Monkey Love, and most recently a primate memoir, which describes his own interrelations with the baboons of Kenya. And he's a brilliant speaker, as anyone who's heard him knows, and he's been described by that authoritative organ of science, the New York Times, as having a style which is somewhat of a cross between Jane Goodall and a Borscht Belt comedian. And I think you'll see that we're in for a great deal of entertainment today from that. Dr. Sapolsky has received numerous awards, among the most important of which I think are the MacArthur Genius Award, and in 2007, the John P. McGovern Award of the American Association for the Advancement of Science. He's also the only scientist I know who's had a rock song named after him by the heavy metal band Johnny Truant. So it's a great pleasure that I ask Robert Sapolsky to come up here and speak today in the Modern Issues in Modern Biology series founded by the Storer Life Sciences Endowment, Stress and Health, From Molecules to Society.
Well, thank you, Ted. It's very nice being here, not least of which because I'm not jet lagged, so that actually as a drawback takes away an excuse if I'm incoherent here. But let me start off. I begin with an assumption, which is that nobody in this room right now has a bad case of leprosy. And correct me if I'm wrong. I'm also assuming hardly anybody here is going to be running off to the bathroom every 10 minutes because of their dysentery. I'm guessing probably no one in this room has liver parasites the size of your fists. And in that regard, we are not like normal mammals. We don't get sick like normal mammals. We don't die like normal mammals. Run-of-the-mill, off-the-rack mammal, what happens? You drink some contaminated water, and 2 days later, you're dead from dehydration. What do? We spend 70, 80 years having our bodies go to hell on us. So oh great, I got to listen to that for the next hour. This is wonderful news. With the exception of TB, HIV, we don't worry about the infectious diseases. Poor nutrition, diseases of poor hygiene, mostly banished. Instead, what we have are diseases where we live well enough and long enough to slowly fall apart over time. And this is a very different realm of disease, realm of who is healthy and who is sick. This is a world in which you have to ask questions like, what your liver does with cholesterol and what your blood vessels are built like and, all the reductive biological questions. But in making sense of diseases like heart disease and metabolic syndrome, all the ones that do us in these days, you have to ask utterly bizarre questions from the standpoint of medical history. Questions like, what's your psychological makeup? Or what's your status in your society? Or how do people with your status get treated in your society? Questions like, do you stop taking your medicine once it starts working? Or even something like, are you the sort of person who eats more when you're feeling unloved? Figure that one out and you have solved half the cases of diabetes in this country. This is a very different world of medicine we have entered into. And when you look at the diseases that do us in these days, we with our westernized lifestyles, these are predominantly diseases that can be caused by or be made worse by stress. And most of us in this room will have the profound westernized luxury of dropping dead of a stress-related disease someday. Now, amid that wonderful news, nonetheless, it's probably a good thing if that happens later rather than sooner, and thus this is important to learn about. And what I'll focus on today is, by now, a 30-year-old literature suggesting that not only can stress do bad things to your stomach walls and your blood pressure and your gonads and all sorts of stuff like that, but that stress and one class of stress hormones can also damage your nervous system. What I'll show you is what we understand to be the cell molecular biology these days of how that works, and most disturbingly, that this is not just relevant to a bunch of neurons in a petri dish. Increasingly, this is clear that it is relevant to the human brain. So at that point, if you're still awake, you should be depressed as hell, because there's nothing but bad news from now until then. That will be the first third of the talk. The last two-thirds will be room for optimism. One at a very reductive level, what could you do? And this will be the realm, as I'll show, of neuronal gene therapy. The other, instead, at a far more integrative level, asking the question of why do some of us get stressed and others not for the same external experience? Okay, so starting off. Whoa, that's okay. Start. It's coming. Wow, that's kind of cool. I've never seen something do like that before. Is that enough, or should we all sit here and wait for more? It's there, okay. I don't know what that was, but starting off, when talking about stress, not just some generic undefined state, but instead stress, the secretion of a single class of hormones. Everybody knows what the main stress hormone is, adrenaline, epinephrine. That is a tragic bit of misconception. Instead, by far the most important hormone of stress, a hormone that I love deeply, a class of steroid hormones called glucocorticoids coming out of your adrenals. Human version, hydrocortisone, cortisol, rodent version, corticosterone, part of the bad news to come, a mere 16 million prescriptions written each year in this country for synthetic glucocorticoids like prednisone, dexamethasone. Standard neuroendocrine cascade, something stressful happens, within 2 seconds your hypothalamus is releasing some hormones, which within 5 seconds, your pituitary thus releases a hormone, and then within a minute or two, your adrenals start secreting the glucocorticoids. These are the hormones that are bad news. And this 30-year-old literature showing that glucocorticoids can be damaging to the nervous system, to this day, sit down an endocrinologist and tell them about that, and they're going to think you're crazy. For the very simple reason that if you don't have these hormones, you're dead. They are essential for life. And thus this question being, how can these hormones essential for life damage your nervous system? That seems rather surprising. The way to begin to appreciate that is to look at what glucocorticoids do. And what they do is shown on the left is everything you would want to have happen to your body if you were being stressed like a normal mammal, like somebody is chasing you, very intent on predating you. What glucocorticoids do is they mobilize energy from storage sites to your exercising muscle. They help to increase your blood pressure, heart rate, deliver that glucose oxygen to your thigh muscles in 2 seconds instead of 3, more likely to survive. Glucocorticoids shut down all sorts of long-term building projects, digestion, growth, reproduction, immune surveillance, and the logic there is obvious. You were running for your life, there's a lion 2 steps behind you, like ovulate some other time. Don't do it right now. Hit puberty next week, grow antlers some other day, a hallmark of glucocorticoid action, deferring what is not essential until later if there is a later. Finally, dramatic foreshadowing here, glucocorticoids get into the brain where they have very dramatic effects in preferentially, the hippocampus, short-term enhancing cognition. So all of that is great, highly adaptive for surviving the sort of stressors that real mammals do, a short-term physical crisis. Where you begin to see the adverse effects of glucocorticoids is on the right, when you have invented the primate capacity to be chronically stressed for psychosocial reasons. And what you see there is the role of glucocorticoid excess in bringing about all sorts of the leading pathologies of westernized life. So you get this double-edged sword. If you're going to be stressed like a normal mammal, you would better secrete these hormones. If you're going to be stressed like a cognitively sophisticated human, glucocorticoids in excess are majorly bad news. Now, it's in that context that we could appreciate this last line, this ability of chronic glucocorticoid exposure to damage the nervous system. And what we now understand is this involves an array of effects. First, glucocorticoids, short-term, as I mentioned, enhancing cognition. Once it is chronic stress, like 8 hours long, you are decreasing glucose, oxygen delivery to the brain. You are screwing up the sort of cognition your hippocampus specializes in. As no doubt an underpinning of that, you are beginning to impair LTP, long-term potentiation in the hippocampus, a building block of what is probably the synaptic version of learning and memory. You are causing atrophy of dendritic processes, disconnecting circuits. Those of you who are neuroscientists know probably the biggest revolution in the field in the last decade, the discovery that the adult brain makes new neurons in the hippocampus mostly in response to all sorts of logical things like stimulation, enrichment, exercise, and the surest ways to shut down neurogenesis, stress, and glucocorticoids. Finally, the bottom 2 lines that I'll focus a lot on, the ability of glucocorticoids to make neurons more fragile, less able to survive all sorts of neurological insults, and at an extreme, their capacity to actually kill neurons. Okay, so looking at this, why the hippocampus and why the dead neurons? Oh, thanks. Why that? And this goes back to the 1960s, Bruce McEwen and colleagues at Rockefeller mapping the first hormone receptors in the brain, apparently totally randomly coin toss. They started with glucocorticoids, and out popped autoradiograms like this, screaming glucocorticoid receptors in the hippocampus. From that day on, if you're going to think about stress and the brain, you're going to think about the hippocampus. And what emerged subsequently was a literature, and each one of these slides is a dozen years' work from various labs, showing lots of stress, lots of glucocorticoids, and you make the hippocampus get older faster. More dead neurons, more reactive gliosis, more memory problems. Conversely, get rid of the glucocorticoids long term, and you slow down the hippocampal aging. So thus, where are these hormones getting off killing your hippocampal neurons? And in the mid-'80s, it struck me that, in fact, glucocorticoids don't need to be neurotoxic. They don't need to be killing the neurons. All they need to be doing is messing them up a little bit. All they need to be doing is making the neurons a little bit miserable, putting them on the edge of a cliff. At that point, if nothing else happens, and neurons recover, everyone lives happily ever after. On the other hand, at that point, if you push the neuron with something else, it's that much more likely to go over the edge. Now, what this idea of endangerment predicted was all sorts of neurological insults, the damage of the hippocampus, should be made more damaging when there's been a lot of glucocorticoids on board. And this turns out to be the case with a vengeance, a whole array of some of the most common insults to the hippocampus out there, ischemic insults, stroke, sustained seizure, oxygen radical exposure, the glycoprotein from the AIDS virus, what is probably the rogue protein responsible for hippocampal neuron death and Alzheimer's disease. All of these, whatever bad things they do to a hippocampal neuron, made worse by glucocorticoids. And the key point here is this is a hell of a lot of different ways to do in a neuron. Whatever this cliff of vulnerability is about, it's a very generalized one. So thus you begin to look at this and say, well, what are the glucocorticoids doing? What are they doing? They could be doing a gazillion things from that chart I just showed you. They could be changing cerebral perfusion rates or blood insulin levels or who knows what, all sorts of peripheral effects. And the best way to show it's not peripheral effects is to do it in vitro and the same exact things there. And using in vitro tissue culture systems, you could begin to do your basic endocrinology. Do you see endangerment in cultures from the cortex, from the hypothalamus, from the striatum? Yes, but not as extreme as in the hippocampus. Do you see endangerment with other steroids, androgens, progestins? Not at all. And very relevant in about 30 minutes, not only does estrogen not make things worse, estrogen makes things better. We'll get back to that. Finally, for those of you who care about such minutia, there's two types of receptors for glucocorticoids. It's the GR that mediates the damaging effects. This is a hormone working in a classical way through its receptor system, making the target cell more fragile no matter what it is you throw at it. So thinking about it, and this was sort of the late 80s, thinking about, well, whatever this cliff is, it is a very generalized one. All of these insults, despite being how heterogeneous they all ultimately have one thing in common, which is they all eventually constitute an energy crisis. With severe enough of hypoglycemia, cardiac arrest, ischemia, all of those, you're impairing the capacity to generate energy. With severe, profound epilepsy, you are outstripping your ability to match energy generation with demands. All of these are disorders involving catastrophic drops in energy stores. All of these are energetic in nature. So looking at this, it struck me that perhaps this cliff of vulnerability was in some way an energetic one. Glucocorticoids in some way energetically messing up neurons. Now, if this was the case, it generated a very simple experimental prediction. You should be able to take some insult that kills like this many neurons, now insult plus glucocorticoids, and you kill this many neurons, insult plus glucocorticoids, and feed the neurons something extra. And you should be able to subtract out the endangerment. And that's precisely what you wind up seeing. One version of this, as shown here, insult alone. This is an insult that mimics severe hypoglycemia. This many dead neurons, insult plus glucocorticoids, as shown. Insult glucocorticoids plus mannose. Mannose, it's this monosaccharide. It's got its own transport system in the brain. Away goes the endangerment. Do the same thing with fructose. Fruit sugar doesn't work, doesn't get past the blood-brain barrier. Use ketones, that works. It's an energetic endangerment. So what are glucocorticoids doing to endanger in this way? And by the early 90s, we got the answer to this. And as neurobiologists, we were thrilled because we were the first ones to see this. And as endocrinologists, we were totally bored because people had known this for 50 years. And it had something to do with what you want to do with circulating glucose when you're under stress. You've just mobilized all that energy from your fat, your liver. You've dumped all that glucose in your bloodstream while you're running for your life. What would be the stupidest thing on earth now to do with your circulating glucose? Store it away in your fat cells for next spring. Store it away in your liver. Store it in your gonads. Store it anywhere. And for 50 years, it's been known that glucocorticoids inhibit glucose uptake and storage in tissues throughout the body except for exercising muscle, diverting it where it's needed and not going into the details, but the molecular mechanisms are understood. So we went and looked, and the exact same thing in the hippocampus. Not a big effect. In a fat cell, glucocorticoids are knocking out like 80% of glucose transport. In a neuron, maybe 20%. That's not enough to kill the neuron, but what it's enough to do is make the neuron a little bit unhappy. And then along comes an insult. So what happens at that point? When I put up here this magnificent 5,000-year-old Egyptian hieroglyphic showing the glutamatergic synapse, this is central to neuron death during these insults, the release of glutamate, excitatory amino acid neurotransmitters, when at excessively high levels, overstimulates its receptors, most notoriously the NMDA receptor, tidal waves of calcium, and at that point you blow apart the cytoskeleton, oxygen radicals, etc., and you got a dead neuron. Critically, every step in this pathway, which is central to the neuron death of hypoxia, ischemia, seizure, hypoglycemia, etc., every step in this pathway is made worse if you're running out of energy. Presynaptic neuron depolarizes more readily, dumps more glutamate, and can't afford to do the high affinity reuptake, sticks around longer, more calcium, can't afford to pump things out. And at this point, you don't need to be a brain scientist to come up with this hypothesis that, oh, insofar as glucocorticoids in an energy-dependent manner make all of these insults that are mediated by this energy-sensitive pathway, maybe what's going on is glucocorticoids, by inhibiting glucose uptake, is screwing up every single step, making neurons less capable of each step there. And I will skip over all the 1990s in my lab showing that that's precisely what you wind up seeing. All of the steps in the cascade are made worse by glucocorticoids, and all in an energy-dependent manner. Give more energy available, and the glucocorticoid endangerment is taken away. Neurons lose control over glutamate, lose control over calcium during these insults more readily. Okay, so in some ways that stands as sort of how we understand the pathways at this point. Two elaborations. One as follows. Neurons do not just sit there getting blown out of the water by these insults. Instead, there's all sorts of defenses they mobilize. Heat shock proteins, upregulation of antioxidants, etc. There's a whole bunch of these defenses. Collectively, they could be reasonably protective. Collectively, they are majorly expensive. And thus, you know what the next sentence is going to be. Not only do glucocorticoids, by way of their effects on glucose transport, not only do they make the insults more insulting, they impair the neuronal defenses against them. So that's bad news. One additional piece of bad news. Most of you who are of clinical background will be outraged by how all of this can't be true because of the well-known fact that, number one, inflammation in the brain after these insults is a bad thing. Number two, glucocorticoids are anti-inflammatory. They should be protecting. And for years, right around this point, which was always the last paragraph in the grant, I would wave my hands wildly and say, okay, well, insofar as, glucocorticoids are doing bad things to the neurons, but good things to the inflammation, so you got arrows going in two opposite directions, you put them together and you ask which arrow is stronger, and if at the end of the day you got more dead neurons because of glucocorticoids, it tells you the endangering arrow was the more powerful one. So about a half dozen years ago, I got my first immunologist in my lab, and I thought it would be a good idea to finally explicitly show the size of these arrows. And what he got instead was a result that now appears to be being found in quite a few labs, a very surprising finding. Under all sorts of circumstances, not only are glucocorticoids not anti-inflammatory in the injured brain, they become pro-inflammatory. And we have seen this now in the hippocampus and even more so in the cortex at the level of inflammatory cell migration, both local and circulating inflammatory cells to the injury site, at the level of pro-inflammatory cytokine expression and secretion, at the level of activation of pro-inflammatory transcription factors like NF-kappa B, at the level of the whole signal transduction pathway. And this is hugely interesting to us on two levels. Number one, a really cool cell biology problem, because in the hippocampus and cortex, in the absence of an insult, glucocorticoids, classically anti-inflammatory. In the hypothalamus, no insult, anti-inflammatory. Injury, hypothalamus, anti-inflammatory. Hippocampus and cortex, injury, and somehow it switches and goes the opposite way. And we think we're getting the answer to that one. We believe we're seeing that there is oxidative damage to a glucocorticoid receptor cofactor that changes its interactions with NF-kappa B. So that's really interesting on that level. The other level at which it's interesting is the gazillion prescriptions written each year to try to block neuroinflammation with glucocorticoids, suggesting that may not be the most clever thing in the world. And the best neurologists for decades have been saying, hey, you notice, while glucocorticoids are great for brain tumor inflammation, they're not so hot for concussive head trauma or ischemic stroke damage. Maybe you should use something else. And for decades, as far as I can tell, neurologists at the less than leading medical institutions in this country continue to marinate their patients in glucocorticoids after something like post-stroke edema emerges. So that's not good news. Okay, what more bad news? Let me just skip over these. What more bad news? Because there's, so much coming. So now we say, okay, this is not good news in terms of the rat hippocampus and the cultured hippocampal neuron. What about us? What about the primate hippocampus? And primates have hippocampi, although they can't spell them correctly, but nonetheless they have hippocampi, and up to a number of years ago, the sole evidence in the field that this might be relevant to us were some studies from my lab using non-human primates showing lots of stress, lots of glucocorticoids, surgically implanting glucocorticoid secreting pellets in the hippocampus of vervet monkeys and coming back a year later, and you get the exact same pattern of damage. This may be in the ballpark of the human hippocampus. And in the years since, what has become clear is it is not just in the ballpark, it is extremely pertinent to the human hippocampus. A number of clinical domains. First one, Cushing's disease, Cushing syndrome, any of a number of tumors where you produce boatloads of glucocorticoids. You get every pathology on the right side of that chart I showed you, and has been known for half a century. You get memory problems, Cushingoid dementia, hippocampal-dependent declarative memory problems, and researchers initially, University of Michigan, doing brain imaging on Cushingoid patients showing loss of volume, atrophy in the hippocampus, only the hippocampus. The more elevated the glucocorticoid levels, the more atrophy, the more memory problems. Is this due to dead neurons? Almost certainly not. Because what they've also shown is go in and surgically get rid of the tumor, and over the next year, the hippocampus comes back to normal size. The next two cases do not appear to be reversible. First one, history of severe trauma producing PTSD, post-traumatic stress disorder, combat trauma, childhood abuse. By now, 25 studies from various groups showing atrophy of the hippocampus, predominantly the hippocampus. The more severe the trauma, the more hippocampal atrophy. Is this reversible? To date, it appears not to be, because these are in folks decades after Vietnam, decades after their childhood abuse. A huge incendiary issue in that field over which people happily stab each other is how much is this the case that the trauma causes hippocampal shrinkage? How much is it the case that a small hippocampus makes you more vulnerable to PTSD? There's evidence for both. I think very, very clearly, though, most of the evidence favors the trauma can cause atrophy of the hippocampus. And fortunately, we're going to know the answer soon, because the wise folks at the Department of Defense did all sorts of brain imaging on a whole bunch of Army Rangers before shipping them over to Iraq, with the high certitude that a fair number of them were going to come back in baskets in terms of PTSD, so they will be able to have done before and after, and probably even get a good publication without having to do more replications. Okay, third model, the one most prevalent to 15 to 20% of us in this room, those of us destined to have a major clinical depression, the archetypal disorder in psychiatry of stress. About half of clinical depressives, highly elevated glucocorticoid levels, and by now a replicated literature showing atrophy of the hippocampus, preferentially the hippocampus. The more prolonged the depression history, the more atrophy, the more memory problems. Is this one reversible? Most of the studies at this stage suggest that it is not. Up to decades after the depressions have gotten under control pharmacologically, the volume loss is still there. Okay, so as promised, bad news. Let me make this even worse. Okay, so there's not only all this, there's not only that issue of all of those folks getting glucocorticoids after their head injuries. What about the rest of those 16 million prescriptions each year for glucocorticoids? Most of those are completely benign. Hydrocortisone for your poison ivy, your swollen elbow, kids taking nasal inhalant steroids for their asthma. None of these are ways of getting a lot of glucocorticoids into the hippocampus. Nonetheless, there are hundreds of thousands of people on high-dose glucocorticoids long-term for their lupus, their rheumatoid arthritis. Do they get memory problems? Yes. It is called steroid dementia, hippocampal-dependent memory problems, and the first good case control studies are now coming out showing for the same severity of the inflammatory, the autoimmune disease, the more it's been treated over the years with steroidal rather than non-steroidal anti-inflammatories, the more memory problems. So one final piece of bad news there, which is you go and have one of these neurological insults and you don't even need your neurologist to give you more glucocorticoids to get into trouble. You secrete so much of the stuff on your own that that adds to the damage. Take a person who has just had a concussive head trauma, a grand mal seizure, cardiac arrest, and you will see the highest levels of glucocorticoids you could find in a human except for somebody with a whole body burn. And it adds to the damage because in rodents, get rid of the glucocorticoids right after insults like those and you decrease the amount of damage. In other words, the typical damage that you get in the hippocampus after some of these insults is damage made worse by the fact that your body stupidly goes and has a stress response at the time. I mean, look at how bizarre this is. Running for your life, glucocorticoids divert energy to your thigh muscles, saves your life. Psychological stress. You're on a blind date. Blind date, you secrete glucocorticoids to divert energy to your thigh muscles. Probably irrelevant. Stroke, you have a stroke and you divert energy to your thigh muscles, completely maladaptive. So what can you do about this? Obviously, you can't adrenalectomize somebody just because they've had a seizure. Nonetheless, there are a lot of things that can be done. There are a bunch of drugs that have been FDA approved for decades, used for other reasons in endocrine clinics, that will shut down glucocorticoid synthesis for up to 48 hours in people. First studies, my lab and others, showing block the glucocorticoid secretion, you could decrease the damage. But what if you're too late for that and you've already secreted the glucocorticoids? Obvious pharmacological strategy, go block the glucocorticoid receptors. Is there a good receptor antagonist out there? Yes, indeed. The famous RU486, the abortion drug, very good at blocking progesterone receptors in the uterus, just as good at blocking glucocorticoid receptors in the brain. A literature now emerging showing the protective potential of that. But what if you're too late for that and it's already bound to the receptor? What I showed you before, bolster the energetics of the system. Because essentially what I showed you, what that whole literature has shown is glucocorticoids are doing nothing more dramatic than making a neuron a little bit nauseous and a little bit lightheaded and a little bit queasy and a little bit energy deprived, and along comes the worst day of that neuron's life. No wonder it's that much more likely to keel over. So buffer things energetically. But what if you're too late for that and you've already messed up energetics enough so that glutamate levels are getting too high, calcium too high, and you have now entered the central arena of neuron death? And what I put here is the segue to the next part of the talk: go and build yourself a better neuron. And this is what my lab transitioned to about a decade ago, going, and in this case, using gene therapy approaches to go and try to make your neurons stronger, more likely to survive neurological insults, more protected against the effects of glucocorticoids and stress. So what can you do with that? Starting off, it is not very hard to go get yourself a shopping list as to what genes you would want to overexpress in a nervous system that has just been damaged. What has always been the difficulty is how are you going to get those genes expressing? Because the sort of gene therapy techniques that are so easy in your liver that like high school kids do it for like Boy Scout projects, none of those work in the nervous system. Most dramatically because neurons are mitotic, and most of the viral vectors used to deliver genes for therapy outside the brain are retroviruses taking advantage of cell division. So what one has to use for neuronal gene therapy are instead the small number of neurotropic viruses, viruses that could do their things in post-mitotic cells, and my lab has exploited various of these over the years, overexpressing genes with the potential to be neuroprotective. Now, just to give you a sense, 30 seconds on sort of herpes, vectorology, forget it, it's not that interesting. Okay, so, oh my God, it's 2 years of work of somebody in my lab. Don't tell them. Okay, so hurtling ahead. So what would you want to show with work like this if you were delivering a gene that's meant to be protective? What's the very first gene we started with back when? It makes perfect sense given what I've been showing you, overexpressing the glucose transporter. Because no matter what the insult is, the neuron's probably going to be a little bit more robust if it's got more energy on board. So put in a viral vector, overexpressing it, and what you first want to make sure, of course, is that the vector is safe. You want to do your molecular homework. Is it expressing? Is the protein winding up in the right place, etc. Next thing you want to show is if you're overexpressing a glucose transporter, you better be able to show there's more glucose transport. And you can show this in vitro, in vivo, won't take you through the details, but showing that this is now a hippocampus taking up more glucose. So if you've gotten that far, the next thing you'd better be able to show is, thus, these are neurons that are in better shape metabolically. And here's a technique called silicon microphysiometry and cultured cells metabolism in real time. Control cultures, throw in an insult, metabolism crashes. Overexpress glucose transporter, maintain much longer. You are bolstering the neurons energetically. So if you've gotten that far, the next thing you better be able to show is these are neurons that are better at handling those tidal waves of glutamate and calcium, and yes, indeed, control or reporter gene only, but glucose transporter, less glutamate in the synapse, less calcium in the postsynaptic cytosol. So if you've gotten this far, the next thing you better be able to show is fewer dead neurons, and you can show this in vitro, and you can show this in vivo. Here's neurons in the dentate gyrus of the hippocampus expressing the reporter gene along with the glucose transporter, showing the more neurons you infect, the smaller the lesion afterward. You can protect neurons this way. So that's one particular question, one additional one, which is critical, built around the fact that most of us are not going to be able to schedule our strokes for next Tuesday afternoon when the doctor has an opening. For the most part, if this is ever going to be done, it's going to have to be after the fact. What sort of post-insult windows of opportunity are there? And you go and you study this, you introduce the vector at the time the insult, you decrease damage. You first introduce it 30 minutes later, 1 hour later, this is glucose transporter in a stroke model. 4 hours later, too late, the window of opportunity is closed. Just figuring out how much time you've got to work with. So using this approach over the years, we have now looked at a gazillion different genes going up and down all the various steps of neuron death. Which in vitro insults do they protect against? Which in vivo insults do we understand how they're working? Do we understand that? Do we understand the windows of opportunity? How many hours do we have to overexpress this gene or that? All of those in hand. A few years ago, we finally began to think about, well, this is great for dead neurons. How about starting to think now about anti-glucocorticoid gene therapy, anti-stress gene therapy? And we transitioned to it at that point. Let me jump over a bunch of things here. To do so, we had to take advantage of how glucocorticoids work. And as alluded to already, there are glucocorticoid receptors. They are cytosolic. Glucocorticoid, the hormone, enters, binds to the receptor, and at that point, the receptor translocates to the DNA where it binds to a glucocorticoid responsive element. Promoter there, turn on genes. Thus, steroid receptors like glucocorticoid receptors are transcription factors. They're hormone-activated transcription factors. So this is what we took advantage of. Around this time, there was a big mystery lurking in the herpes virology field, something we probably all know about. You get yourself a herpes infection, you get a cold sore, whatever, it goes away, and then a month later, 50 years later, it reactivates. It comes out of latency. When does herpes reactivate? When you're under stress. Bereavement in humans, social subordination in rodents, chemotherapy, all of those. Why does this make sense? It's a great time. Stress, you're immunosuppressed. Suddenly herpes comes out, goes through a whole bunch of replications, and goes and hides again. So the question always was, how does this little piece of viral DNA know that the host organism is miserable? And a Harvard group got the answer to this a few years ago. Herpes measures your glucocorticoid levels. The herpes genome is studded with glucocorticoid responsive elements upstream of all the genes involved in reactivation out of latency. So we took one look at this and wet our pants. We were so excited, and we immediately took all our vectors and filled them up with glucocorticoid response elements, and we put half a dozen of them in a row and all of that. And the idea was to build ourselves vectors that were stress-inducible, glucocorticoid inducible. And as shown here, they are inducible by glucocorticoids, natural or synthetic, not the other steroids, in a dose-responsive manner. Here's another hieroglyphic. In this case, showing the higher the circulating levels of glucocorticoids, the more expression. Or in this case, a rat undergoing a seizure and inducing a great deal of expression. This is building a smoke detector into your neurons. Along comes a stressful period, and that triggers the expression of these vectors. So with that trick in hand, we could then begin to build vectors meant to block the effects of glucocorticoids. And to do so, we got a whole bunch of families of genes, one of them expressing an enzyme that breaks down glucocorticoids. That's a pretty good anti-glucocorticoid strategy. Another one overexpressing a dominant negative glucocorticoid receptor, that as well. The third one is my favorite, and it takes advantage of what I mentioned about 20 minutes ago. The fact that not only does estrogen not make neurons more fragile, estrogen is neuroprotective. You go and have an acute neurological insult and you want to be dipped in a vat of estrogen. It is a very strong antioxidant. It induces trophic factor expression, and estrogen, just like glucocorticoids, similar mechanism of action, estrogen binding to its receptor, which translocates to an estrogen responsive element. And thus what we did was we made ourselves an artificial gene that was half of a glucocorticoid receptor and half of an estrogen receptor. The hormone binding domain came from the glucocorticoid receptor. The DNA binding domain came from the estrogen receptor. What's the result of this? Stressful period, glucocorticoids bind to this chimeric receptor, which then translocates to estrogen responsive elements. In other words, diverting this endangering river into instead a protective one. And various ways showing that this winds up working. Here's but one example. BDNF, for those of you not oriented to BDNF is a good thing. You want to have a lot of BDNF. Glucocorticoids make BDNF levels go down. Estrogen makes BDNF go up in the hippocampus. So here we see treating rats with estrogen and up go BDNF levels. Here we have a rat with this chimeric estrogen receptor, glucocorticoid receptor gene exposed to a neurological insult, and the glucocorticoids now work like estrogen and boost up the BDNF levels. So using this approach, we've now begun to explore some other domains. Instead of trying to do gene therapy against neurological insults, beginning to think about gene therapy for rat models of psychiatric disorders. And to just very, very briefly touch on this, in this case, shifting over to the amygdala. Those of you who think about the amygdala, central to fear conditioning, anxiety, the amygdala in people with PTSD. The amygdala very sensitive to glucocorticoid actions. So here, overexpressing some of these anti-glucocorticoid genes in the amygdala, and you are blocking aspects of fear conditioning. You can block aspects of anxiety. I'm just blasting over these here. You can block some of the morphological changes that occur in amygdaloid neurons during chronic stress. All of this possibly suggesting we could learn something about psychiatric disorders with tools like these. Finally, in this domain, forget about gene therapy for neurological diseases or psychiatric diseases. You could now begin to use these anti-stress vectors to make a hippocampus that works normal than better. Can you make a hippocampus that is improved during chronic stress? And lots of different ways of showing this. Just to jump over very quickly here, it is now possible to get a hippocampus which during stress with neurogenesis makes more neurons rather than less neurons during stress. It is possible to begin to manipulate the systems this way. Okay, so that's great news. That's wonderful news. The next slide I've been putting up for about 12 years now, and for the first couple of years it was just being incredibly pleased with myself putting it up. And now I put it up as just a major embarrassment. What this is this is an explant of human brain tissue. This came out of somebody with a tumor where this was some cortex, and we got it 30 seconds later. And you'll see the outline of the blue there, little bits of reporter gene from herpes viral vectors we were able to introduce there. These vectors will express in human brain tissue. And a dozen years later, this is the best data picture we've ever gotten. It's not ready for the clinical world yet. It is not going to be at the neighborhood grocer soon. All sorts of stuff needs to be modified if neuronal gene therapy of any of the sorts being contemplated will ever be useful for acute neurological insults. So some good news there, but nonetheless stay tuned. Okay. Second realm of some good news. Back to that chart I put up before, what could be done, block glucocorticoid secretion, block the receptors, more energy, make better neurons, all of that. There was another thing put up on that could be put up on the chart there that is so idiotic and fatuously obvious that I couldn't even bear to put it up, which is don't get stressed. Oh, cool. That's really clever. Don't get stressed. I've just had a concussive head trauma. Let me psychologically reframe my experience. Whole realms of stressors where you ain't going to be doing anything to decrease the psychological impact of it, but most of us are not getting stressed in that way. Most of us are not running from predators. Most of us have psychological stressors. And transitioning into this zone from the very first day that people began to study stress as a concept in the 1930s, what was very clear from the start with the first rats was individual variability. Some organisms are more vulnerable to stress, and thus a critical question becomes, once we consider psychological, psychosocial stressors instead of concussive head traumas, once you move into that realm, why do some bodies and some psyches deal better with stress than others? And that's what the last part of this talk is about. Okay, so you want to understand individual differences in this realm. Individual differences, vulnerability to psychogenic stress. Who are you going to study it in? You can study it in humans, and there's really interesting stuff out there, findings like major clinical depression, and for years afterward, you've increased your risk of heart disease, and that is absolutely solid by now, but God help you if you need to figure out the intervening steps in the physiology there. It is incredibly hard to do. You outlive your thesis committee. Seeing the downstream consequences, very hard to do. Okay, so how about you study this in rats? Shorter lived, you can control the stressors, you control the pathologies, you examine afterward, that's great. There's one problem though, which is if a rat is a good model for your psychosocial stressors, you've got problems. So, taking something in between, how about non-human primates? Obvious advantages there and obvious problems that could potentially come in dealing with captive primates if the housing is too small, if there's all sorts of things there that distort behavior. And fortunately, you guys are in the neighborhood on the campus of one of the best primate centers in the country, but some of the places where people study captive primates, you are not studying models for human behavior, you are studying models for human imprisonment. So that could be easily distorted. So over these years, as Ted alluded to, over these years, for the last 30 years, each summer, I skip out of my lab to go and study these issues, individual differences and vulnerability to stress-related disease in a non-human primate population, but one in its own natural setting. And studying olive baboons, savanna baboons. These are my study subjects. If there's any way to pull the light down, probably better contrast, or does anybody know which button it is? And if they do, okay, go for it, Ted. Whoa. Oh, just hit them all. See what happens. Ted, you didn't stand a chance. It's like up to the fifth button here. Up. Okay. Well, okay, I'll just pantomime baboon violence. Okay, so study subjects are olive baboons, savanna baboons, and these are the I've studied over the years, classic savanna primates living in these groups, 50, 100, 150 animals or so. This is my study site, the Serengeti of East Africa, and what I'm interested in, this is the perfect place on Earth to study for a very simple reason. If you are a baboon, this is where you want to live. It's a great ecosystem. The baboons only spend 3 hours a day getting their days cooked. They're in these big organized troops, so the lions don't mess with them very often. Their infant mortality rate is lower than amongst the neighboring humans. Critical implication, which is if you only have to work 3 hours a day for your daily calories, you've got 9 hours of free time every day to devote to making some other baboon just miserable. And that's the critical thing. We're not getting our ulcers because we're being chased by predators. We're getting our ulcers from members of our own species for psychosocial reasons. The baboons are just like that also. Out there, nature, bloody in tooth and claw, they're one of the exceptions. Overwhelmingly, if you were a baboon in the Serengeti and you were miserable, it's because another baboon has worked very hard to bring this state about. They are all about psychological stress. They are great models for us. So what is the psychological stress about? You begin to look at these guys, and within days, what is clear is not all baboons are equal. In fact, there are dominance hierarchies in number 1, number 10, and number 20, and the quality of your life varies dramatically as a function of your rank. What does that do for you? At least, that's not going to do it. Close the doors or something. There's no shades there. Okay, it's really a drag. Okay, what you would see here is baboons grooming each other. Your social rank has something to do with who grooms who. Your social rank has something to do with, that comes through. Okay, your social rank has something to do, is one allowed to show this in Central Valley? Okay, your social rank, Bay Area, snotty guy. Okay, your social rank has something to do with your reproductive success. Do you get to be in the safest spot in the tree when the predator's around? Do you get to sit in the shade? Your dominance rank is very, very important. Here's an example. Here's a male who's just predated an impala. This took a lot of work. This is very hot stuff for this guy. And along instead comes the alpha male in the troop, and our guy picks up, walks away, and number 1 winds up with a kill there, exploiting the labors of the working class. And what's most striking in that case is the guy who did the work, he leaves without a fight, without a nasty word under his breath. These guys are smart enough to understand where they are in the hierarchy. It's a very stabilizing, very conservative feature of their lives. So you look at these guys and you're mighty impressed with rank, and thus you begin to ask, what goes into being a high-ranking baboon? And for reasons I could explain later, about 90% of the work I've done over the years have been with the male baboons. What goes into being a high-ranking male? One critical thing is winning key fights. Male baboons have canines that are bigger than you find in male lions. They use them. This guy, can't quite see, has a canine slash in his shoulder. And these are not just ritualized fights. This is a male who had joined a troop of mine about 2 weeks before, and the only way to describe this guy is he had horrible political skills. He was hassling all sorts of guys he should not have been going near. One evening, coalition formed against him, and this is what was left in the morning. And in the 31 years I've studied baboons, the leading cause of death of male baboons are male baboons. So it is not just symbolic violence, it is highly, highly real stuff. Far more important than violence, though, are threats of violence. Conventionalized gestures, a male here, for example, displaying his canines to the guy in the next tree, in effect saying, look, you remember what happened last week? I've got the same teeth I had then. Do us both a favor. Don't push your luck. And most of the time, what you get instead of a fight is a ritualized subordinate gesture from this guy. Now, if all these guys were about was either violence or threats of violence, they wouldn't be very interesting. What they're most doing are things that are much closer to home to us, things that are much, much more psychologically stressful. So just to blast through this, what is it that makes psychological stress stressful? And a huge, elegant literature by now shows that for the same external misery, you will subjectively feel more stressed, you will turn on more of a stress response, and You are more likely to get a stress-related disease if you feel like you have no outlets for the frustration caused by the stressor, if you feel like you have no control over the stressor, if you feel like you have no predictive information as to when is it coming, how bad is it going to be, how long is it going to last, and if you feel like you have nobody's shoulder to cry on, if you lack social support. And what these baboons are brilliant at is manipulating these psychological variables. First example. Here's a male who is in a sexual consortship with this female. She is ovulating in estrus. He is grooming her. He has been with her for the last 3 days. And you will notice, just happening to be 6 feet away on the other side of the bush, is another high-ranking male who just happens to have been 6 feet away around the clock for the last 3 days. Never an inch closer, never overtly threatening, just around, just harassing the consortship. And this is enormously costly. This guy hasn't eaten in days, he hasn't slept, and what you wind up seeing is 40% of the time, without so much as a threat gesture going on, this guy will pick up and relinquish the consortship to this guy. This is not violence, this is threats of violence, this is grinding psychological stress, and these guys are good at it. Next example. Here's one of the wild card strategies. If you're a male baboon, get yourself a coalitional partner to back you up in a fight. And it is not trivial doing something like this. Highly conventionalized gestures built around essentially saying, I'm not pissed at you or you pissed at me. No, in fact, we're both getting along with each other, but we're really pissed at him, so why don't we? And this triangulating stuff, very difficult to do. And what we've got here are two of these coalitions facing off against each other. And if you don't know baboon body language, these are 4 uptight baboons. Violence is about to break out, and a second later, yes, indeed, they start fighting. And anybody notice the 4th baboon? Let's see, where'd he go? Oh, there he is. There he is, showing this principle of our close relatives, which is when the going gets tough, bail out of there. Almost half the time when the fight occurs, your partner skips out on you. And of the times that he does, a third of the time, he joins the other team. Talk about psychological stress. You're going into a fight that's potentially life-threatening, and you don't even know who's on which side. Lack of control, lack of predictability. Final example. Male baboon in a consortship with this female, in this case being threatened, harassed by this guy, high intensity from only a few feet away. This guy in the middle is under staggering pressure there. You know that he's about to crack, violence is about to occur, and a second later, yes indeed, he does become violent. What does he do? He spins around and he bites the female. Another feature of this close relative of ours, which is when the going gets tough, look for somebody smaller to take it out on. Half of baboon violence is displacement aggression. Male loses a fight, chases a subadult, who lunges at a female, who bites a juvenile, who slaps an infant. What does that wind up meaning? You're sitting there minding your own business, bird watching, and somebody else is having a bad day, and it's your rear end that gets slashed. Lack of control, lack of predictability, lack of outlets. So looking at all this, what goes into being a successful high-ranking male? Yes, being prime-aged and big and sharp canines and muscular and all of that, but all sorts of stuff that would have to be grouped under the rubric of social intelligence, which coalitions to form, which ones not, impulse control, which provocations to walk away from. And starting in the late 70s when I began with these guys, something else that struck me was high-ranking guys would be better at dealing with stress. And this is what I set about trying to do, to see if there was a relationship between social rank and the physiological stress response in these guys. And what I'll show you is I proceeded to waste my next 15 years looking at that, because there's something much more interesting going on, much more familiar with what we are about. Okay, so how do you study this? Part of it is you do your basic Jane Goodall scene and collect behavioral data, but what you also need to do is get physiological data out of these guys. You need to anesthetize them. This comes with all sorts of constraints. Number one, you got to find an anesthetic that doesn't mess up the hormones you're looking at. Number two, you got to do everybody at the same time of day to control for circadian rhythms. Number three, if you want to get a first blood sample that tells you something about nice basal non-stress levels of these hormones, you can't dart somebody if they're sick, if they're injured, if mated, if they're had a fight, if you want to measure cholesterol levels, you can't dart them if they've had breakfast yet, all those constraints. Next, if you want to get a first sample that's nice and basal, you can't dart somebody if he knows it's coming. In other words, you can't like get in your Jeep and chase the guy back and forth for hours, and when he's wounded, you can help him. And what I do, this was a PBS thing some years ago, a frame where you may be able to see a white flash on this guy's shoulders. That was the instance of a blowgun dart hitting him, injecting the perfect darting, and that up until a second before, he had been looking at a Kenyan field assistant of mine, no anticipatory stress. Final constraint is, if you want to get a first blood sample telling you about basal levels of steroid hormones, you've only got about a minute between the time that the guy goes down to get the first sample. So given all those constraints, about once a decade it works flawlessly with an anesthetized baboon, where you can take tissue biopsies and CSF fluid, blood samples. And last year I was doing EKGs on these guys. You can have heparin locks on your acacia branches, really high-tech stuff. Guy recovers overnight in a cage. Next morning you release him back to his buddies there. His samples processed on a hand crank centrifuge, which is as awful as it sounds. Get your dry ice shipment once a week or so. Keep your samples frozen, sneak them through customs, and you're all set. Okay, so given all this, what was I going to look at? Obvious what I was going to look at first with my obsession. What's the relationship between rank and glucocorticoid levels like in these guys? And by now, you should design the perfect glucocorticoid profile, which is, in the absence of stress, you want really, really low levels. Along comes a psychological stressor. Don't get suckered in by it. Keep the same. Along comes a real physical stressor, have the biggest, fastest response possible, and the second it's over with, have the fastest recovery possible. And this is precisely what you see in the dominant animals. If you are subordinate, your resting levels of glucocorticoids are elevated, because even under non-stress circumstances, you're stressed. You sit there and you spend 10 minutes digging some tuber out of the ground, and anybody can rip you off. You spend the morning somebody to groom you, anybody else can disrupt it. Somebody else is having a bad day, you get slashed, lack control, predictability, outlets. When a real physical stressor does come along, the subordinate males have a much sluggish, more sluggish response to the stressor, and when it's over with, they have the more sluggish recovery. So exactly what you don't want to have. And I proceeded to spend the next dozen years working my way up and down this neuroendocrine system in these guys, asking, okay, so if you have more circulating glucocorticoids in the subordinates than the dominants, what's working differently at the level of the brain, pituitary, adrenals? I will spare you the details and give you the punchline. If you are a low-ranking baboon, you have elevated glucocorticoid levels mechanistically for the same reasons that you see elevated glucocorticoid levels in people with clinical depression. CRF, blunted pituitary sensitivity to it, impaired feedback, dexamethasone resistance. If you don't know this jargon, don't worry. The main point being that a subordinate baboon is like a human depressive in this realm. And what is the soundbite about depression in cognitive psychology? Depression is helplessness. And in lots of ways, if you're a subordinate baboon, what you need to do is learn to be helpless. So with this sort of approach, okay, I'll just blast through these. With this approach, over the years, working my way up and down these various physiological systems, and you always see the same punchline, which is there will be differences as a function of rank, and it's always the subordinate animals with a profile that is more predisposing towards stress-related disease. So this seems to give a really clear take-home message, which is if you get a choice in the matter, you want to be high-ranking and go out and compete and win, and bludgeon the other guy, and big social Darwinism blowout. Let me take the last couple of minutes to show you how this is idiotically simplistic, how there's much more interesting stuff happening. Yes, rank is important, but far more important is what the rank means in your society. One of many different examples here. All the data that I've shown you come from stable dominance hierarchies. What do I mean by a stable hierarchy? Number 5 is defeating number 6 95% of the time. Number 5 is losing to number 4 95% of the time. On the other hand, if number 5 is winning only 51% of the time with number 6, that's an unstable dyad, and they may be just about to flip. All of the data I've shown you are coming from stable hierarchies, more than 90% of the interactions reinforcing the status quo. Every now and then, though, once a decade, something happens, a critical guy dies, is injured, coalition forms, whatever, where for about 3 months, the whole hierarchy becomes stable. The highest ranking guys are still higher ranking than the most subordinate guys, but amid the upper ranking cohort, rank is shifting every 3 hours. Everybody's fighting, injuries, nobody's eating, nobody's mating, nobody's grooming. And what you see here, during a stable period, nice low resting corticoid levels in the dominant males. During an unstable period, elevated. It's not being high ranking, it's what high rank means in your primate society. And in this society, it means you're having to your rank once every 2 hours. In a stable hierarchy, high-ranking males go a year without having a fight. It's not the rank, it's what the rank means. Next one, not just rank, not just its meaning, but personal experience of both. Let me give you one example of this. This was a number of years ago, and an extremely aggressive male joined the troop. And this guy not only was aggressive, but had the really charming trait of just attacking all the females. And about 2 weeks after he joined the troop, I darted a whole bunch of the females. And he had come in the middle of the season where I had darted a bunch of females before. And what I was able to do after 2 weeks was keep track how many times had this guy attacked a female to the point of drawing blood. Then I looked at the white blood cell counts in the female. And what you see here is a dose-response curve. The more often a female was attacked by this guy, the more immunosuppressed she was. Here were the females in the troop before he joined. No attacks at all, and there's no difference. So you ask, well, what are the effects of having an aggressive, destabilizing male on immune function in female primates? And the answer is, it depends. It depends on what it means to you. If you're sitting on the sidelines, it doesn't mean anything at all. It's not the abstract state of being in a violent society. It's how often is your own personal nose being rubbed in it. So not just rank, not just the social meaning of it, but how it plays out for you. Final variable, and most important one, which is your personality. Saying the word personality in a primate talk used to cost you tenure, but now it's one of the two trendiest terms in the field, the other being culture, non-human primate culture. Non-human primates have stable temperamental differences which qualify as personality. They differ among male baboons, how often they start their fights, do they like to groom, do they play with infants, etc. So a number of years ago, I sat down and went through 15 years of behavioral data on these guys and tried to pull out as many personality markers as I could think of, and I anthropomorphized like crazy. What's a baboon's equivalent of having a hobby? What's a baboon's equivalent of whistling on the bright side and walking on the sunny side of the street? Came up with like 50 variables, controlled rank, controlled stability of rank, and out came a very interesting finding, which is all the great physiology you get if you are a high-ranking male has nothing to do with being high-ranking. It's due to the sort of personality that's associated with high-ranking males. And you could be the highest-ranking guy on the block, and if you don't have these personality markers, you're going to have the same rotten physiology as number 20 in the hierarchy. It's not rank, it's personality. And thus the final question here is, what sort of personality works for a male baboon? Coming from the Bay Area, like I could spend my weekends in Marin giving hot tub seminars to baboons, telling them how to do their stress management, because this is exactly what you would tell them to do. How do you go about your male-male competition? I will spare you the details and just say, you are a male baboon. Can you tell the difference behaviorally between your worst rival on the planet threatening you in your face versus taking a nap at the other side of the field? If the guy is threatening you in your face, do you at least grab some control and start the inevitable fight? If you have a fight, can you behaviorally distinguish between whether you've won or lost? And if you've lost, do you have an effective coping mechanism? Do you go and groom with somebody else? Do you go and beat up on somebody else? Or do you go and vote by yourself? Beating on somebody else or grooming with somebody else are both equally effective. What you wind up seeing, oh great, that's wonderful news. In other words, what this is built around is, can you tell the difference between the big things and the little things? If it is a big thing, can you exert some control? Can you tell if the outcome is good or bad? And if the outcome is bad, do you at least have some effective coping mechanisms? People pay to be taught those things. And you go back in the records and you look at the guys with these low cortisol traits, as I've been showing there, these guys with these low cortisol traits, and you go back in the record, and when they were juveniles, that's exactly what they were like then, and they outlive the high cortisol cohort by 2 to 3 years. So concluding here, starting off with terrible news. We are now 60, 70 years into dealing with the concept that stress can do bad things to every outpost in your body. We have to begin seriously to consider that stress can play a role in the most common of neurological disasters that do us in, and almost certainly having something to do with why some of us will age cognitively more successfully than others. And what is also clear is that it's almost certainly relevant to our own brains. At the reductive level, we are beginning to understand what goes wrong enough that there's the chance of rational interventions that may translate into something clinically useful at some point. And finally, shifting to the other end, when we deal with the stressors that plague our neurotic middle-class life, which rarely involve saber-toothed tigers and rarely involve grand mal seizures or concussive head traumas, we transition to the stressors that all of us mostly deal with. The level of intervention is not going to be at the genome, and it's not going to be at the level. It's not going to be at the it's going to be at the level of the psychological viewpoints that filter whether or not we view the external world as stressful. So there is some room for good news there. So on that note, let me first acknowledge the members of my lab and acknowledge my field collaborators over the years, including my wife. Okay, so thank you.
Want to answer some questions? Well, Robert has kindly agreed to spend a little time with you answering questions if you have any.
Okay. Anyone want me to take out their adrenals? Okay, any questions? Okay, great. Very, very interesting point. Glucocorticoids actually increase appetite and play a role in the craving for comfort food during stress. CSF has done incredibly cool work showing precisely how glucocorticoids are working at neuropeptide Y, etc., in the hypothalamus to increase carbohydrate craving. Wait a second, you're running for your life. Why should you be craving laundry starch at that point? It's time course. CRH, that hypothalamic stress hormone, is coming out within seconds. CRH is an anorexogen. CRH decreases appetite. Glucocorticoids, coming out far later, stimulate appetite. So by definition, if only CRH levels have risen, you're in the first couple of minutes of the stress response. You want to decrease appetite. By definition, if both CRH and glucocorticoid levels are up, you're right in the middle of a stressor. Put the two pieces together, CRH is more powerful. Appetite. By definition, CRH has a 5-second half-life, glucocorticoid a 90-second one. I'm sorry, a 90-minute one in humans. By definition, if glucocorticoid levels are high and CRH is not, you're not in the stressful period, you're in the post-stressor by now, and what glucocorticoid stimulating you to put energy back to the places you mobilized it from. So glucocorticoids increase appetite in that sustained profile. They increase fat deposition in that sustained scenario, and they preferentially do it to abdominal fat. So it's an issue of time course. Let's see. You had a question? Okay. How does exercise fit into all of this? Exercise is great news insofar as it stimulates neurogenesis and BDNF, various trophic factors. Exercise is good news insofar as it does great things for your cardiovascular system, etc. It does great things for people who find it to be pleasurable, and that's a whole literature in terms of stress management techniques. If this is a type of stress management technique that is not quite your favorite one on earth, it's not going to do much for you. For example, a very solid literature shows some of the capacities of meditation to decrease blood pressure, things like that. If I were to meditate 20 minutes a day, I would have a stroke by this week. It is so antithetical to what I'm about. Nonetheless, it does wonders for lots of people. Individual differences, blah, blah. It's got to be for people who actually find exercise to be stress-reducing. Final element shown in a super elegant study by Liz Gould at Princeton, there's got to be an element of control. Take a lab rat who is choosing to run a running wheel, and you run lots there, and as shown, you increase neurogenesis. Now, psychology jargon, yoke that running wheel to a running wheel in the next cage. There's a rat in there which is kept inside the running wheel. It can't get out, and every time this one rotates as well. They're both getting the exact same exercise. This one is doing it voluntarily. This one is being forced. This one increases neurogenesis. This one decreases it. So the control element, the feedback, the pleasurable aspects can completely override some of the biology of it in some cases. Let's see, more questions. Yes?
In your panel with the 3 slides about depression and PTSD patients, that we know that the hippocampus shrink when you have depression, PTSD, severe trauma, etc. My question is, has anybody looked to see whether a combination of exercise with the depressants can actually reverse that?
I'm not sure. There's been one study so far showing antidepressants can reverse the volume loss. There's been 5 or 6 showing that they don't work. As far as I know, nobody has explicitly coupled that with exercise. That is pertinent, though, insofar as not looking at people who do sort of major sort of marathon type exercise. There's a very elevated incidence of depression amongst the population. Other jocks have a much lower level of depression and neuroticism than one expects in the general population, as we all learned in high school. But people who are like long-distance runners have very elevated levels of depression, often elevated levels of glucocorticoids. Classic interpretation, often the long-distance running is self-medicating the depression. So one could certainly speculate that would be part of it. As far as I know, it hasn't been. Let's see, more questions. Yep, in the back.
Yeah, could you comment a little bit on the relationship between sort of a hypocortisol response that you see in some patients with post-traumatic stress disorder and autoimmune disorders like idiopathic urticaria, rheumatoid arthritis, and that sort of thing?
Okay. Trying to figure out how to herd the cats in this subject. Another huge controversy in the PTSD field is whether PTSD patients have elevated glucocorticoid levels or lower than normal levels. That has been highly controversial. I would say, even though I don't like it, the majority of studies show lower than normal levels. However, a very disproportionate share of the studies have come from one laboratory. And that work has not always been replicated. Almost certainly it's the ever reliable, there's heterogeneous subtypes that explain it. Nonetheless, lower than normal levels. Wait a second, lower than normal levels, what are you doing atrophying the hippocampus with lower than normal levels? What the same groups have shown is enhanced target tissue sensitivity to those glucocorticoids. So, at the end of the day, it may work out that there's more of a hit there. It's unclear. Nonetheless, the strongest evidence suggesting that the hippocampal atrophy is a consequence of the trauma is the fact that you look at kids who have had childhood trauma and they have normal hippocampal size. It's not until late adolescence that the atrophy begins to emerge. Thus, it cannot be the model that having a small hippocampus before an insult is the one that predisposes you towards PTSD, because these kids have normal hippocampal volumes. It is a time bomb that goes off as a consequence of the stressor. So that's been hugely controversial. In terms of the other half, autoimmune disorders related to glucocorticoids. So what everybody always thought about over the years is that what glucocorticoids do is, they're immunosuppressive to the extent that it could really be bad and make you sick, psychoneuroimmunology. What was soon recognized when some of the assays became more sensitive in the 90s is that very early on in the stress response, what happens is you enhance immunity, and that's mostly driven by catecholamines. At that point, Alan Monk at Dartmouth had this wonderful insight, which is what glucocorticoids are doing to the immune system during stress is not suppressing you below normal levels. What they're doing is bringing you back down to normal afterward. It's only in the face of really pathological chronic stress that you not only but you crash even lower. What he predicted based on that is if you fail to secrete glucocorticoids under that situation, the immune activation fails to be countered by glucocorticoids and just gets more and more extreme. He made the prediction that a variety of autoimmune disorders would be associated with hypoactivity of the adrenocortical axis, and that has been amply confirmed since then. So within the normal physiological realm, glucocorticoids are not immunosuppressive. They are meant to take an activated immune system during a stressor, and that makes a certain amount of sense. If your innards are about to be ripped out by a predator and dragged in the dust, that might be a good time to sort of fire up the immune system. Glucocorticoids are bringing you back to baseline. It's in the pharmacological world that they are immunosuppressive. Bet you didn't think I was going to download at that rate. Okay, any more questions? Or yes? Oh, there are two of them. One is the issue of the viral vector being safe. The second is the issue of delivery. Currently, pretty much the only way you could do stuff, you can artificially open up the blood-brain barrier, and you can deliver vectors that way. You can have none of these work very well. And even more importantly, all of those are not specific, just releasing the circulation all over the brain rather than where they're needed. Currently, about the only way you could do it is drill some holes in the head there and deliver it directly by infusion. And a handful of clinical trials for gene therapy done with humans, a couple of Alzheimer's ones, some Parkinson ones, have been done with that approach. That's not going to work because in the realm of head injury, stroke, etc., because you're not going to wrestle down the person as soon as they come in the ER and drill holes in their head. That urgency business, it's never going to work that way. In addition, an awful lot of the neurological insults will be spread across a whole bunch of tissue. You're not going to inject 11D times out there, even if you could do it quickly enough. The challenge to the field is going to be safe delivery, so that you're doing it to all the places where there's damage and not to any of the other places. And that has completely stumped the field at this point. And basically all of us doing this at this stage, if that one doesn't get solved in the next decade, we've spent a decade having all of our papers and grant proposals at the end talking about the glowing promise of clinical gene therapy in the nervous system. If that one doesn't get solved, it's going to run out of steam.
Join me in thanking Dr. Sapolsky for his wonderful talk.